Okay, many thanks to the organizers for organizing this year after year. It's a really nice place to sort of meet old friends and see new work. I'm going to talk about some work done by two students of mine and a couple of collaborators on bacteriophage, which are viruses that infect and attack bacteria. So for those of you who haven't seen these creatures before, they look like something that's come out of a science fiction horror movie, and they kind of behave like that too. This is a picture of a lab situation where one bacterium has been attacked by many, many viruses. But in fact, even a single virus particle, if it infects a bacterium, can create a situation like this that you see here. It takes over the bacterium and its machinery, and it produces, hundreds of copies of itself. Eventually, this bacterium bursts and dies, and about 100 or several hundred new offspring viruses are produced. So this fact makes it a kind of very unusual predator-prey system. You can imagine, suppose you had a fox eating a single rabbit, and imagine that after eating a single rabbit, it produced 100 new foxes. You could quite legitimately ask, why don't the rabbits get wiped out? Why don't bacteria get wiped out by viruses like these? And there are many reasons. There are lots of attack and defense mechanisms that these bacteria and viruses have against each other. But one reason why this wiping out doesn't happen is because there's a large class of viruses that have a certain amount of self-restraint. And these so-called temperate bacteriophage, when they infect bacteria, they can go into two modes, either this death mode where they just make lots of copies and kill the cell, or they can go dormant. And they live more or less happily ever after inside the bacterial cell. When the bacterial cell divides, they divide. And this is good for the bacterial cell, not just because it doesn't die, but also because it gets immunity to further infection from the same type of virus or similar viruses. So this is one reason why this kind of, these kind of viruses and bacteria are able to coexist. And I've been very interested in this decision because very little is known about this. People have done some experiments by putting bacteria and viruses together in a test tube and seeing what happens. And they noticed that for a very, very wide range of virus bacteria pairs, from the soil, from marine ecosystems, from the gut of animals, you find that the percentage of time an infection goes towards dormancy is a very narrow range, around 10%. And nobody really understands why this number is 10%. Is it just a historical accident, or is there some evolutionary advantage to being in this range? Or conversely, is there some detriment to a virus having a 50%, say, dormancy rate? These are the kinds of questions we're interested in. So the only argument that's been put forth so far about this number and why it might be in this range is to say that these things live in very complex environments. And in some cases, big catastrophes, some chemical might appear which kills off the viruses. But in other kinds of situations, maybe some other catastrophe happens, some other situations arise which kill off the bacteria. And in this kind of uncertain situation, it's good to hedge your bets and have some in this state and some in that state. And in this kind of argument, the dormancy percentage would be set by the relative frequency of this kind of environment versus that kind of environment, and the kind of intensities and frequencies of these catastrophes. So while this is undoubtedly part of the explanation, we decided to approach this from a slightly different angle, which is to think about competition. So in all these bacteria virus ecosystems, there are many, many species around. It's never just one bacterium and one bacteriophage. There are many species. So we wanted to ask what happens if viruses are competing with other kinds of viruses for the same pool of bacteria. So we set up a little toy model where, let's say, there are two types of viruses, both competing for the same pool of bacteria. And they're identical except for the way they do this dormancy decision. So let's say 1% say goes 10% dormant, one goes 20%. And we set up a simple set of equations, very much like Lotka-Volterra equations. I'll be happy to tell you in more detail about them later. And we run this. And this is very typically what happens. If you follow the bacterial numbers, they start off growing initially exponentially. They're hardly affected by the viruses because the virus numbers are very, very low. But over this build-up time, viruses do build up. And suddenly, at some point, they really start shooting up very fast, at which point the bacterial numbers really crash. Everything gets infected in a very short time. After that, what happens is the only thing left are these dormant bacteria. either of type 1 or type 2, and they grow and they reach some steady state. And on these kind of timescales, there is no reversal back from the dormancy back to the death mode. So it reaches a nice steady state where some part of the population is of one type and some part is of the other type. So now you see, we can think of this as from a game theoretic point of view. So this becomes like a two-player zero-sum symmetric game. The two players are the two types of viruses. Winning in this game simply means in the final steady state, which one had more proportion in the population, which virus sort of took over most of the population. And the strategy space is this dormancy percentage. So, are you playing with 10% or 20% or 100%? These are the strategies. So from this kind of game theory point of view, what you can do is you can play every strategy against every other strategy pairwise and build up a payoff matrix, which tells you how much does one strategy win over the other and by what amount. So this is what a matrix like this looks like. These are the two different strategies, two different players playing different kinds of percentages of dormancy. And this is the sort of payoff matrix, who won and by how much. And when you have a matrix like this, you can ask, is there a best strategy? There are many ways to define a best strategy, but one way is, if there is a saddle point, it defines a particular kind of equilibrium point, where if this was an evolutionary game and one player was playing this strategy, there would be no incentive for the other player to move away. Okay, so we do in fact find that there is such a thing, and it's around 10%. And I'll cut a long story short. We do understand why there is an optimum like this. Different parts of the dynamics push the strategy in different directions. During the crash, it's very important to go dormant so that you build up a little population here that can finally take over. But in the beginning, it's important to go into the death mode so you can build up enough things, enough of your population that can actually infect lots of bacteria in this stage. So these are pulls and pressures in opposite direction, and you find an optimum somewhere in between. But the interesting thing really is not that there's an optimum, but that if we vary parameters, now here I've shown you various different parameters in this model, and we're able to vary them by huge orders of magnitude, well outside even biologically reasonable ranges. And we find in all cases, except for one parameter, the optimal is very narrowly in this 5 to 10, 15% range. Okay. Again, there's not enough time to really take you through all the details, but we understand analytically under reasonable approximations why this robustness is there to parameters. We understand the kinds of trade-offs between different aspects of the dynamics, which is what essentially leaves this optimal to be extremely robust. So at this point, given, if you believe me on this robustness, I've presented a somewhat reasonable argument perhaps to convince you that given the fact that we see real bacteria and phage producing this 5 to 10% dormancy, it's quite possible that competition between viruses is one of the important evolutionary selection pressures that is setting this number. But in using this kind of game theory argument of a two-player game and applying it to these experiments where a population of bacteria and viruses are interacting, I'm making a fairly big assumption that all viruses of the same type are actually behaving in the same way. And that's certainly likely not to be true. In fact, even in a simple case where you take a test tube and you throw viruses and bacteria together, there will be many heterogeneities. One very obvious heterogeneity will be that some viruses will not get in, some bacteria will not get infected at all. Some will get infected by 1, some by 2, some by 3, just by pure chance. So one could ask, if a virus is able to now update its strategy and exploit this kind of heterogeneity and perhaps make the dormancy percentage depend on this so-called multiplicity of infection, could it do even better? Thanks. So it turns out, so we took this, we put this into our model, we did the whole game theory again, and we do find there is an optimal strategy. And it turns out to be this very interesting one, where if you are a virus that is infected, a single virus infecting a bacterium, then you should always kill the bacterium and produce lots of copies, lots of offspring. Whereas if you are two have infected or more have infected a bacterium, that bacterium should go dormant, 100%. This strategy beats every other strategy very well, and it does it partly because of what I told you before. What you really want in a good strategy is to be going into the death mode early on, and then when the crash happens, to go into the dormant mode. And somehow this multiplicity of infection in this 1 and 2 switch turns out to be a really good heuristic for detecting when to switch your strategies. Okay, so let me, my time is up. I'll just say one thing that in the 1970s, there's an experiment where people studied this old phage called lambda, and they found that it does exactly this. And it does exactly this when there's a single infection. I won't go into the details of the experiment, but when there's a single infection, it kills the cell. And when there's double or more infections, it goes dormant. Now we found more and more and more examples, which you can find over here, of cases where there are more phage, which we now know do this kind of strategy. So overall, let me just end by saying we think we've given a reasonable argument for the possibility that competition between phage is a rather important thing and produces rather strong evolutionary pressures that set many aspects of how this decision is done. So maybe next time I'll tell you about more aspects of these decisions, but I'll stop now. Thank you.
Why don't the bacteria that choose a lysogenic lifestyle, why don't they just take over wild type altogether?
So in fact, we do find in many natural populations that there are bacterial genomes that have been sequenced have about 5 to 6 of these prophages, of these viral genomes embedded within them. So in fact, I do think a lot of natural isolates of bacteria do in fact have these dormant bacteria inside them. Over time, of course, some of them may get inactivated and so on, and various things may happen. But typically, I think wild bacteria do have lots of these things around.
So we are out of time, and I would invite Sarika to give her talk on synchronization in multiplex networks.